Agriculture has completed one cycle and is turning its phase. Indiscriminate use of pesticides and chemicals during Green Revolution has polluted soil, water, and the whole ecosystem. Now, we find organic farming movement everywhere. The farmers are in search of alternative ways to supply nutrition to crops and to manage pests and diseases. All the creatures on this earth have the right and ability to live and survive. Hence, we need not and cannot eradicate any pest, pathogen or weed. It is enough if they are kept under control to avoid economic damage to our crops. The best way to do this is the integrated pest and disease management system. There are many useful insects and organisms in the nature. There is one or the other natural enemy for all creatures. This brings the balance in the ecosystem. Identifying and employing such natural enemies of the crop pests for their management is the principle of biological control system. Enemy could be fungal pathogen, NPV, predator, or parasite. Use of Entomopathogenic Nematode or EPN is one among them and is the latest addition. EPN represents a group of soil-inhibiting nematodes that parasitize a wide range of insects. It kills the host insect in 24 hours and do not harm any plant. These nematodes belong to two families, Steiner Nematidae and Heterorhabdidae. These nematodes have a mutualistic partnership with anaerobic bacteria and together they kill a wide range of insect species. Xenorhabdus bacteria associate with Steiner nematids and Photorhabdus bacteria with Heterorhabdids. The bacteria are vectored from one insect host to another only by the free-living nematode stage, known as infective juvenile, which lives in the soil. Nematodes entering the insect host release the bacteria into the insect's hemolymph, which kill the insect by massive septicemia. The bacteria degrade the insect's tissues, making it as the food source for the nematodes. It facilitates them to mature and multiply. Usually, 1 or 2 generations of adult nematodes are produced within the insect cadaver. The progeny of the last adult generation reassociates with a few bacterial cells and nurture them in their intestine. Then, the nematodes move out of the insect cadavers into the soil and wait for another insect to parasitize. Since the discovery of entomopathogenic nematode in 1927, efforts are on to employ them against agricultural pests. In the last 2 decades, EPN is successfully included in biological pest management system. NIPHM Hyderabad has developed a novel and low-cost method for EPN mass production and formulation. National Institute of Plant Health Management, Hyderabad, is an autonomous institution under the Department of Agriculture and Cooperation, Ministry of Agriculture and Farmers' Welfare, Government of India.
Our institute is like a pioneer institute which has been doing research training and also some kind of practical orientation in policy formulation by the Government of India. We have been helping the government in imposing quarantines, in studying various biosecurity measures of Government of India. Our institute has developed a package of practices or a manual in respect of each crop, how pests can be controlled in an integrated manner. This institute, with the support of Department of Science and Technology, we have taken up a very interesting project. This is called Entomopathogenic Nematode, or it is otherwise called EPN, or beneficial nematode. And these nematodes, when they are applied in the field, they are very effective on many of the insects. I must appreciate all my scientists, especially Dr. Sunanda, for having worked hard for so many years and to isolate these beneficial nematodes. I am sure these nematodes will be of so much useful to the farmer in controlling many difficult pests which cannot be controlled by pesticides.
The mandate of NIPHM is capacity building by training, consultancy services, and research in the field of plant health management and biosecurity. It promotes environmentally sustainable practices and technologies.
The National Institute of Plant Health Management, which is pioneering a campaign on successful use of biological control for sustainable plant pest management. Large number of predators, parasitoids and biopesticides have been encouraged and they have been successfully cultivated on a low cost production technology to help the farmers to cultivate and produce such materials at their farm level and use efficiently and bring down their input cost in the sustainable production of plants, which are safe for the consumer in the respect of pesticide residues and public health and ecosystem of agriculture. In this endeavor, the project sponsored by the Department of Science and Technology, scientists working in this project successfully developed some of the new protocols and patented protocols for large scale multiplication of entomopathogenic nematodes. I hope this video will help to reach to large number of farmers across the country.
Entomopathogenic nematode can be an important component of biological control system. With fundamental knowledge of nematodes and insects, any plant protection scientist can isolate, culture, store and evaluate against target insect pests. EPN is safe to non-target organisms and environment, but its mass multiplication is cumbersome with conventional laboratory methods. Hence, EPN is not so popular in India. To address this issue, NIPHM took up DST project. Dr. B.S. Sunanda is the principal investigator. Under this project, they have developed and patented two vital low-cost technologies for mass multiplication of EPN. Now it is being popularized through training to technical officers of various organizations and companies.
At National Institute of Plant Health Management, we are working on various groups of nematodes like entomopathogenic nematodes, beneficial nematodes and plant parasitic nematodes, quarantine nematodes. But we are having one DST funded fast track young scientist project which was given by Science, Engineering and Research Board in New Delhi with the grant size of 17.4 lakh rupees. So, we are more focusing on beneficial nematodes for the management of any insect pests, but we are focusing mainly on soil dwelling insect pests.
Now, let us study the techniques of isolation, mass multiplication, storage and usage of entomopathogenic nematodes in detail. It is the very purpose of this video presentation. To tell you in short, EPN is isolated from the soil, inoculated to the host insect, harvested from the insect cadaver and stored in different formulations for further usage. Soil sample collection. Select moist area of the field between the rows of plants or irrigation channel. Dig a pit of 15 cm depth. Collect 5 random samples. Put the soil sample in a plastic bag. Note the date of sampling, place and associated vegetation, etc. on a slip and keep it with the sample. Collect such multiple samples from different locations or crop fields. Rearing of greater wax moth or Galeria melonella. We need to produce large number of wax moth larvae for EPN baiting from soil and for further multiplication. Collect natural honeybee comb. Cut the honeycomb into smaller pieces and place it in a plastic jar with lid. Find and separate fully grown larvae and pupae of greater wax moth. Put larvae, pupae or egg cards into the jar. Keep it under room temperature for the development of wax moth larvae in large numbers. These larvae can be used for EPN baiting or for further mass production. If natural honeycomb is not available, greater wax moth can be reared on artificial food. The dietary composition is like this: corn meal 400 gram, wheat flour 200 gram, wheat bran 200 gram, milk powder 200 gram, yeast tablets 100 gram, honey 400 ml and glycerine 300 ml. Mix the contents to make paste like this. Keep this food in a plastic box and put wax moth eggs or larvae for multiplication. Take out the larvae, retaining few for further multiplication. Life cycle of greater wax moth is like this: eggs 5 to 10 days, larvae 20 days, pupae 8 days, adults 3 to 5 days, and fecundity is 300 to 1000 numbers. Rearing of rice moth or Coursera cephalonica. In the similar way, rice moth is also used for baiting or multiplication of entomopathogenic nematodes. Mass production of rice moth larvae is simple. Break sorghum grains and keep it in hot air oven to kill any insect or pathogen. After cooling, keep it in a plastic tub and mix rice moth eggs. Cover the tub with cloth and keep it for development of rice moth larvae. Take out necessary number of larvae for usage and retain few for pupation and emergence of adults. Collect the adult moths and keep them in plastic funnel of this size. Cover the mouth with a sieve cloth and the nose with a cotton swab. Feed the adult moths with 10% sugar solution in a cotton plug. Keep on collecting the eggs regularly. Sieve it to separate eggs from debris and store the eggs in a glass vial for further usage. Isolation of nematode from soil. Insect baiting technique. Remove any debris like wood or bark pieces, leaves, etc. from the soil sample to avoid contamination with saprophytic microorganisms. Spray water to maintain moisture if necessary to facilitate the movement of nematodes. Put 3 to 5 bait insect larvae and spread the soil on it. let the lid has few pores for aeration. Keep the container in dark at room temperature at 22 to 25 degrees centigrade. Take out the dead insect larvae after 2 to 3 days. Cadavers with brown coloration are usually parasitized by stainer nematodes, whereas brick red to dark purple are parasitized by heterorhabdids. Recovery of nematodes from infected cadavers. White trap. The dead cadavers are rinsed with sterile water twice to clean up soil and dirt. Set the size of circular Whatman filter paper to a convex watch glass. Moisten the paper with sterile water and place it like this in a petri dish of bigger size. Put the cadavers on the filter paper with a little distance between them for easy movement of emerging infective juveniles. Pour 20 ml of sterile water into the petri dish so that it keeps the filter paper moist always. Cover the petri dish with the lid and label it with species of nematode and date of inoculation. Keep the white trap in room temperature or in BOD for 4 days for emergence of infective juvenile. To complete one life cycle, it takes between 7 to 9 days in case of stainer nematodes and 9 to 11 days for heterorhabdids. Infective juveniles come out of the mouth part of the dead cadavers. Harvest the infective juveniles into a beaker. Put sterile water again to the white trap for further harvest of infective juveniles. Add more water to the beaker and allow nematodes to settle at the bottom. Then carefully decant the supernatant water. Repeat this for 2-3 times until the water is clean. Place the nematode suspension in a tissue culture flask at a concentration of 1000 to 3000 nematodes per ml of water. Store the flask in a cold room or in incubator between 10 to 20 degrees centigrade. Check the stored flask periodically as the shelf life of EPN is variable. Usually, stainer nematodes can be stored for 6 to 12 months without the need of subculturing, whereas heterorhabdids may require more periodic subculturing. In vivo rearing of entomopathogenic nematodes. Further multiplication of EPN can be done in a similar way. Place Whatman filter paper in a petri dish. Evenly distribute 1 ml of infective juvenile suspension at the rate of 1000 to 2000 nematodes per ml on the filter paper. Add 10 last installed larvae of greater wax moth to the dish. Here, we are providing approximately 100 to 200 infective juveniles per larvae. Cover the petri dish with the top and label it with date, species, etc. Place the dish inside a loosely sealed plastic bag to conserve moisture. Keep it in dark at room temperature or in incubator at 20 to 25 degrees centigrade. After 3 to 5 days, remove the cadavers with the sign of nematode infection. Transfer the same to a white trap to harvest infective juveniles. If the cadavers smell putrid, it may indicate contamination and failure of nematode infection. Storage of nematode or formulation. Nematode can be stored with optimum moisture, temperature and oxygen. Aqueous suspension form of storage is sufficient for small-scale or lab purpose. Adjust the density of nematode to 10,000 infective juveniles per ml. It should be aerated using aquarium pump thrice in a week and change the water every week. Nematode can be stored in talc-based formulation, sodium gel alginate capsules and sponge formulation. Keep all these formulations packed in polythene bags to prevent desiccation. Keep verifying the viability of infective juveniles in periodic intervals. Application of EPN. It is better to carry various formulations of EPN in a cooler box to the field. Put aqueous suspension or sponge with infective juveniles into water in a bucket. Squeeze the sponge to release the nematodes. Spray it with a suitable sprayer to the crops for above-ground insect pests. It is better to take up spraying in cool evening hours to facilitate effective infection of insect larvae by infective juveniles. Spray the solution to the soil around the plant base for soil-borne insect pests. In another method, infected cadavers are mixed with sterilized vermicompost, fine sand or cocoa peat and applied to the plant base.
We have conducted a number of field trials by using entomopathogenic nematodes in the farmer field, especially in Sangli district and Kolhapur districts in Maharashtra. I have applied nearly 20,000 of infected cadavers of Gelleria melonella with a 4th instar larvae. When we have broadcasted with the vermicompost and a cocoa pit, we have got a very good success, nearly 70 to 80% of the root grub management, specifically in the sugarcane crop. So I would like to definitely say my heartfelt thank you to ACRB DST New Delhi and letting me to implement this project at NIPHM with all kind of facilities.
Entomopathogenic nematodes are successfully used on various crop pests such as pod borer, cutworm and diamondback moth on vegetables, root grub of sugarcane and betel nut, etc. NIPHM has demonstrated effective control of sugarcane root grub and other soil-borne insect pests in farmers' field of Maharashtra. NIPHM conducts training for State Agricultural University and ICAR scientists, extension officials of state departments and farmers. It also conducts trainings for officials of various organizations and companies. Training is to create awareness about the application of EPN in biological pest control, isolation and mass multiplication of EPN for large-scale usage, etc.
Presently, we are using existing technology for mass production of entomopathogenic nematodes, but it's not possible for a field application. That is a white trap method for extracting nematodes. But we have designed some technology by using low cost materials. By this patented technology, we can mass produce nematodes in a large scale or at a farm level, we can say, with very less cost and not much labor or not much any sophisticated equipments or BODs like that. This patented technology we are ready to give for any interested parties from corporate sectors or any government agencies or SAUs or ICR people can adopt this technology by learning or by getting the training from us at NIPHM based on the cost.
NIPHM sells its patented technique of mass production and formulation. The innovators of this patented technology are Dr. Korlapati Satyagopal, IAS, ex-Director General NIPHM, and Dr. B.S. Sunanda, Assistant Scientific Officer, Nematology. The unique features of this invention are easy to adapt, low-cost technology for mass production of EPN, requirement of limited space, no need of sophisticated lab and high-cost inputs for both multiplication and preparation of formulation.